Hi everyone. I welcome all of you to the Fusion Talk on behalf of Pre-Science in Silico. I welcome all of you to this talk, and I'll speak some words about Pre-Science in Silico. At Pre-Science in Silico, we team up with both industry and academia to tackle challenges in drug discovery and material science. Apparently, the idea for this Fusion Talk series, it has came from our wish to get experts from different fields to share their valuable insights. And I take an immense proud to tell you that this is the first Indian company to create our own drug discovery software with advanced AI features. Pre-Science in Silico was founded by academicians who wanted to make AI-powered discoveries in drug and materials accessible to everyone and aiming to redefine the landscape of scientific knowledge. And it's my pleasure that we have Professor Dan Major from Bar-Ilan University, who will be our speaker for today. And the title of his talk would be Development and Application of EnzyDock, New Docking Strategies for Enzymes. About Dan Major, he has completed his undergraduate Ph.D. studies from Bar-Ilan University with Bilha Fischer. After his PhD, he's gone to University of Minnesota, where he teamed up with Jiali Gao, where he has done his postdoctoral research, predominantly in the field of development and application of theoretical methods for enzyme catalysis. In 2007, he has joined Bar-Ilan University, Department of Chemistry, and currently he's a professor of chemistry at Bar-Ilan University, and he has published more than 150 papers. His main research interests are in the field of computational chemistry, biochemistry, and renewable energy. And yeah, I would also like to tell that I've been a part of his group, and I've had a very great time working with him, and certainly looking forward to the his very insightful talk. Now I hand over the stage to Professor Major. We are looking forward to the next talk. Yeah, please.
Okay, thank you very much, Anil, for the kind introduction and the opportunity to speak to you here today. It's a great pleasure. And yeah, so I'm excited to share some of our recent stuff. So yeah, so I was speaking about development and application of EnzyDock, and I will speak about new docking strategies for enzymes, which is stuff we've been working on in the last few years. So, but let me start by showing some pictures from 2018, when Anil was in the group. This is, you only see part of the group here in these pictures during a brunch. And so Anil was part of the biosimulations team. And here you see some of the people in the materials team. So we usually have about half the group on biocatalysis and the other half on material science. And we often have great talents coming from India working in our group. So that's been a tradition for many years already. And this coming year, we'll be having 6 out of about 14, 15 people in the group will be coming from India. So I would like to tell you today about a few topics. So first of all, why did we feel there was a need to develop another docking tool? We have so many, so why another one? And then after giving you some of the motivation, I'd like to speak to you about the concepts in EnzyDock, which is multi-step and multi-scale docking. And then give you some examples. One example is ligand sensing in a family of enzymes called terpene synthesis. And then I would like to take you to a work we did in the time of the corona. If you all remember those days, we did a benchmark docking study for the MPRO enzyme in SARS-CoV-2. So let's get started with the motivation. So why do we need another ligand protein docking software? So we were working on an enzyme back in the 2007 or so called nitroalkane oxidase, and it does a simple oxidation of a nitroalkane into nitrite and hydroperoxide. And this is a simple mechanism. You have first an aspartic acid extracting a proton. You get a carb anion here that's formed here, and then this will form an adduct with the cofactor flavine, and then there will be subsequent redox reaction. We were interested really only in the proton abstraction because in those days we were really interested in tunneling effects in enzymes. So we had developed several path integral quantum simulation methods for that. So we were really looking to study simply the proton abstraction step. So a crystal structure was available in 2006, which is more or less when we started the project. And if you notice carefully, here is the adduct where the carb anion has formed a covalent bond with the flavin cofactor, and the nitro group is forming in this direction, kind of out of the active site. And that was kind of puzzling to us because we had thought it should point in the other direction. And we then started the modeling study, and initial docking attempts with the leading docking software at that time, which will go unnamed for now, it went nowhere. We didn't really get any reasonable results that could match with what was expected from catalysis. So the idea with them was, okay, we know how to do docking. It's not difficult. So we said, let's write an independent docking script in Charm. We were using Charm software at the time, which can allow you to do a lot of scripting, and you can do really advanced simulation protocols using Charm. So we said, let's randomly dock the ligand in the active site. Let's do some Monte Carlo and MD simulations. And then we had developed a highly accurate semi-empirical QMMM potential. Today, this would be called a machine learning potential. In those days, we called it a reaction-specific potential. But it was really accurate with DFT level accuracy at a cost of a semi-empirical calculation, which could be up to 1,000 times faster. And then the idea was to, well, let's add some chemically sensible restraints to narrow down the search space. And we use for that purpose nuclear Overhauser effect or NOE restraints, which you can do simply and easily in Charm. And those were put in to keep this interaction in place. And then we were hoping for some consistency and convergence. And that we did get. We did the initial docking, randomly generating starting conformations. And after the docking process, we got a set of consistent poses where this nitro group were all facing in the right direction, forming a hydrogen bond here and here. So that seemed to make sense to us, although we did not agree with the available crystal structure at the time. Well, going with our intuition, we then computed kinetics, that means free energy profiles using QMMM methods, and we calculated isotope effects. And these were all in good agreement with experiments. So here you see a free energy profile or so-called potential of mean force. And here you see a classical profile that's with classical mechanics. And if you add quantum effects with what we call path integral simulations for hydrogen and deuterium, so the ratio between these will give us the isotope effect. Okay, so here we see the calculated numbers, and these were in very good agreement with experiment. This is the experimental value 14, and we got about 16, which is considered good agreement for QMMM. So here's just a picture of the transition state from those simulations. And the kinetic isotope effect was in really good agreement with experiment. These numbers are considered to be excellent agreement, and we got the correct trend from water to the enzyme. And here we also found that there was a significant amount of tunneling, and this was published in PNAS in 2009. But throughout this study, we did not know whether we had the correctly docked pose or not. Well, in 2009, just as we were about to publish the paper, we got a crystal structure from our co-workers, and the crystal structure showed that our docked pose was really in excellent agreement with that found from the crystals. So we could call it beginner's luck, but it showed that going with an incorrect crystal structure would have led us in the totally wrong direction in the entire project, which took a couple of years. So it also essentially stood on the fact that we had predicted the correct pose, and had it been wrong, everything would have been wrong. So here's the X-ray versus the QMMM docking pose. Okay, so the conclusions we drew from that work was that, first of all, it's not trivial to generate the correct initial pose. And the other question is, are we doing enough to assure that we have a good starting point for these enzyme simulations? And as we wrote in this review paper, we conclude this section by stating the obvious yet painful reality. It is difficult to overemphasize the importance of the initial docking step. An incorrect initial Michaelis complex will necessarily lead to inaccuracies in the modeling of the enzymatic process. So essentially, if it's garbage in, then you're going to get garbage out. So we have to be careful about the initial structure. Oh, so that was our motivation, that standard docking tools might not give the correct starting structure for enzyme studies. And that's, in our opinion, was a big problem. So we then thought, well, how can we do something that is can use some of our chemical intuition, some of our knowledge, and allow us to do something we call today multi-step and multi-scale docking with EnzyDock. So this has been developed in the group by several researchers. The main developer today is Renan Na, who does all of the coding on this program today together with myself. And let's see how we do this multi-state and multi-scale docking. So here I'm going to give an example of something that is a multi-step reaction or multi-state system. So here we have a system that's a terpene synthase system. So you have a substrate called geranyl diphosphate. So you have a carbon chain connected to a diphosphate. And the chemistry here really isn't all that important to us. We just need to notice that there are many, many chemical steps taking place in one active site. It's multi-step, multi-state. And the question is, can we do this docking correctly? And questions like, which state should we dock? Should we dock this one? Well, this one is highly flexible, so it might be difficult to dock it correctly. Also, being a carbon chain, this one has no hydrogen bonds to the active site. So it's all weak van der Waals interactions. Can we predict those correctly? And then you have charges that hop around in this case. And these charges, these carbocations are stabilized by the active site. Will our docking predict correctly how these are stabilized? And so on. So there are many questions that we would like to answer using our docking. It's not enough just to try get the correct pose, and then if we're lucky, we get it, and if not, then we don't. Because if we don't get the correct pose, then again, studying this entire reaction will be incorrect. Okay, so here are some of the points that I just mentioned. And of course, points like stereochemistry. For chemists among you, stereochemistry is critical. So if the stereochemistry is wrong, then that's also a big problem for the study. Okay, so we have this idea then of multi-state. So let's say we're looking now at this reaction we saw before in a schematic manner. So we have the substrate, intermediates, and let's say the final state here. So what are the things that we know about this system? Well, we know about the initial cleavage site. And if you look at the previous slide, we know where the cleavage takes place. We know about the regiochemistry, that is how the different parts in the molecule approach one another, from which side, let's say the Re or C phase. We know about the stereochemistry of the reaction, if that's characterized well. And in the case I showed you, there is a water doing a nucleophilic attack. We know about where the attack should take place. We might know about the deprotonation site, and that should be near to a base in the active site. And we would assume that intermediates are all stabilized during the catalytic cycle. And then we have a very important concept that we call consensus binding. That is, throughout this reaction, we do not presume that there is significant tumbling or motion of the entire ligand in the active site. There's usually not enough time for that in these types of reactions that are relatively fast. So we assume that all the binding poses should be quite similar. And we have experimental evidence for this. So this is not just our intuition, it's based on experimental findings for many enzyme systems. So we need to assure consensus binding. And that's not at all a given with any docking program. So if you dock all of these states separately using any docking program, including EnzyDock, you might get different opposing poses for each of the states. Okay, so that was the multi-state idea. What's the multi-scale idea? Well, the multi-scale idea is starting as usual docking. We dock on a grid. So the enzyme or the protein is described as a grid. Then we do, following the docking on the grid, we do a full MM scoring. And then we do QMMM scoring for the best poses. That allows us to treat anything between intermediate transition states, which we might want to treat doing docking in enzymes. MM is usually treated by CHARM and CGNFF. This is the ligand-based force field of CHARM. And the QM region, we can use anything between semi-empirical methods and DFT methods. So whatever we want to use, we can use here. So let's look at EnzyDock under the hood. So how we do the different things here, and I hope it won't be too detailed for those of you who are less method inclined, but I will keep it fairly short and brief. So okay, so like any software, EnzyDock needs to prepare the enzyme. That's the protein structure, reading cofactors. That's critical. Not all docking programs can treat a lot of cofactors, so we can treat a lot of cofactors. Water molecules, meaning explicit water molecules, and selected flexible residues, and do the grid calculation. That essentially prepares the enzyme. Then the ligands are prepared either by providing EnzyDock with coordinates or SMILES strings. And then EnzyDock will do simulated annealing, Monte Carlo, and molecular dynamics to create multiple conformations in vacuum. And then these are clustered using torsional clustering to generate representative dominating conformations for docking. And then essentially the docking takes place iteratively on a grid, like any docking program. You can do then Monte Carlo translation and rotations, and then do simulated annealing Monte Carlo on rotatable bonds with simulated annealing molecular dynamics. And then following that, we do Cartesian clustering. And at this stage, this is where we add, or the user can add, consensus restraints, and those are the restraints that assure that within a user desired limitation, the poses will be similar. So you can then enforce restraints so that all poses will be similar to a given template structure. That template can be the first docked pose, or it can be a crystallized inhibitor you already have from a previous crystal structure. And then it will force all the subsequent docked ligands to adopt poses similar to that template. We can also add any kind of distance NOE restraints within the ligand or between the ligand and side chains. You can add dihedral restraints within the ligand or within the enzyme, and additional types of restraints as are available within the CHARM program. And then following Cartesian clustering, we then do full MM minimization outside the grid, meaning a full all atomistic. We can add implicit solvation, and then we do QMMM minimization to get a final scoring. And then in the final step, we then use something called Pathfinder, which helps us find consensus paths. So let me explain this idea of Pathfinder. So here are just some of the main points within EnzyDock. So let's look at this Pathfinder idea. So the idea is to find consistent poses along a reaction coordinates. So let's take the example reaction I showed you just before. If you have the substrate bound in many types or many poses, then you have intermediate A in many poses, intermediate B, C, all in many types of poses. Then the question is, which of these poses here match the poses for A, match the poses for B, and so on. So how do we find that? So Pathfinder then helps us identify matching poses along a reaction coordinate. So let's say this S matches this intermediate A, which matches this intermediate B, and this C, and so on. Okay, so it helps us identify plausible reaction pathways. EnzyDock is not so easy to use from a simple Linux interface, so we have developed together with a team of CharmGUI an interface, and in CharmGUI our EnzyDock is called EnzyDocker, and it allows you to do essentially everything from building the system from an initial PDB structure to selecting flexible residues, defining the grid and doing the entire calculation within CharmGUI. So that's a helpful additional tool that we've put in, and the paper describing this is in preparation at the moment. So applications of EnzyDock. So we looked at it, used it for many different types of systems. So this is something I will speak about towards the end of my talk today. That's the SARS-CoV-2 MPRO enzyme. We did benchmarking of different types of docking software. We used it to look at nerve agent detoxification in phosphotriesterase enzyme. So here you see different type of inhibitors where we mark the chemically similar motifs that EnzyDock can identify automatically using cheminformatic tools. Here you see the docked pose. And we use it for Diels alderases. There are enzymes that do the Diels-Alder reaction, and there are actually quite a few enzymes that have been discovered in this family in recent years, and they do quite complicated chemistry as well. And here we see again parts of the molecule that are deemed similar by EnzyDock, and those are the ones that will be used for consensus docking that will made to adopt similar poses. And here we see sets of docked poses using consensus docking for enzyme called LEPI. So these are some of the basic test cases that we've used to emphasize what EnzyDock can do. Okay, so now you have some basic idea of the ideas behind EnzyDock. Then let's go into depth and look at a system that we worked on recently, and that's terpene synthesis. And we looked specifically at a question of ligand sensing. So this was for us a fascinating project. I think maybe the most exciting project I worked on in recent years because of the things we found, not because it was planned to come out like that. So the people who worked on that was Renana, the main developer of EnzyDock, and Shani, who develops complementary reaction and cheminformatic tools for EnzyDock. And so we call this project Making Sense of Substrate Sensing in Terpene Synthesis, and it was published recently in Angewandte. So let me tell you about terpenes. Terpenes is a fascinating family of molecules. They're formed by terpene synthesis, and they have what we call chemodiversity. So it forms the largest group of natural products in nature. So you can find over 80,000 known compounds that come out of terpenes. So that's huge. Now, many drugs are natural products from the terpene family. So here you see a drug called Taxol, which is an anti-breast cancer agent that are used commonly in the pharmaceutical world. Of course, steroids are all derived from terpenes. And all the smell molecule, or many of the smell molecules in nature, like the smell of lemon, the smell of menthol, come from terpenes. So if you smell something nice in nature, chances are that this is from a terpene. And of course, the unique smell of cannabis is from a multitude of terpenes that together give a very unique smell in cannabis. So terpene synthesis have a range of substrates coming in different sizes, but they all use the same building blocks. And that's what makes this really fascinating. So you have in differing sizes, you have something called GPP, FPP, and GGPP, where you see that this has 10 carbons, 15 carbons, and 20 carbons. Now, each of these, like so many enzymes will bind GPP and form different products. And similarly, many will bind FPP and form many products from this building block. If you look at the active site in these enzymes, it's also fascinating. You have a highly biphasic pocket. When I say biphasic, I mean it has two faces. It has a polar face where you have highly charged and polar residues. And you have a very hydrophobic site. So that's built by strategy by the enzymes, where you have the diphosphate, which is highly polar here. Then you have what we call an isoprenoid cation. So that's a carbon chain with some double bonds, and the cation. And this one is bound, when you see there are a lot of aromatic residues, which stabilize this one by pi cation interactions. This is a common structure for all terpene synthesis. They look like this. And then you note that this diphosphate is bound with 3 magnesium ions. So it's highly polar, but note that you have 2 magnesium hat that are symmetric, and then you have one that's kind of in an asymmetric position right here, okay? And one of the questions that I'm going to raise today is, when this one is bound initially to this oxygen here or here, as you see, there is a CO bond here, here, and here. So when this is initially bound here, we don't know whether it's bound to this one or to this one. Or in other words, we don't know how the enzyme senses the ligand through this oxygen or through this oxygen. That can have critical impact on what happens later on in the catalysis. So let's just give you an overview of terpene synthesis. So we have the C10 family with a small substrate, and it can form many, many types of monoterpenes. These are just 3 of those. Then you have the C15 family, or the sesquiterpenes, and these can form like these, products like these. And then you have the diterpene family, which can form the C20 family. Again, there are many, many of these, and they can form up to 80,000 different products that come out of these terpenes. So it's a huge family, exciting chemistry, and you can do lots and lots of on this playground. So I mentioned that. So our question was, okay, so to which of these 2 oxygens does this carbon tail bind to, here or here? So we then collected a set of a handful of crystal structures where we had a ligand that binds either to this or this. There aren't that many crystal structures. We took what was available, and we started to dock, and we looked at the total docking energy, and here's the PDB ID. And we calculated the docking energy, whether it was bound to O1 alpha, this position, or to O2 alpha binding to this position. And here you see that in this 2O and G, this is a limonene synthase, which gives a lemon smell. There's a strong preference for binding through this oxygen. And the energy we found from docking to the other oxygen was much, much higher. So there's a huge energy difference between the 2 oxygens it binds to. If you look at several others, similar enzymes, they show the same preference. They have a strong preference for one oxygen over the other oxygen. That was fascinating. 1, that there is such a preference. 2, and we know the preference from the X-ray. So we know that this one is preferred, but the docking energy also managed to reproduce this preference. So that was important to us, which shows that EnzyDock can easily differentiate between this and this oxygen binding. And it's interesting that there is this preference in nature. But then we looked at another family of terpene synthesis, and they show the opposite binding preference. So we have 2 classes of binding, and this was not discussed in the literature until this point, and we didn't understand why. Anyway, we are happy that EnzyDock could reproduce this, but when we look more carefully at the enzymes from these 2 families, we come to a fascinating finding that these are all plant enzymes, and these are all microbial enzymes. So they bind differently. Okay, so that was really exciting for us, that there is something systematic here. And when you look more carefully at the differential substrate binding preferences, and you try and analyze what's there in the active site, then we see clearly that there's a set of conserved residues, and these are positively charged residues that coordinate this diphosphate, and they're conserved positions in 3 dimensions, and of course in sequence. If you look at the microbial ones, they also have 3 positively charged residues, but in different positions in space and in sequence. So there are motifs or patterns of binding that are plant-like and microbial-like. So that was fascinating. Then we said, okay, so what are the reasons for this? How does the enzyme then use this to differentiate between binding this way or that way? So we said, let's look at this most simple idea, that's looking at the active site volume ratio. Is there in the plant enzymes bigger volume on this side than on this side that makes it preferentially want to bind here in plants, and the opposite in microbes? So we define the following descriptor. Well, let's calculate the volume ratio, the volume near the one oxygen versus the volume near the other oxygen. And we did this by numerical Monte Carlo simulation, which is essentially Monte Carlo integration, as you will find in standard numerical analysis textbooks. And we calculate this ratio. And what we find is that for all the enzymes where we had the correct answer, meaning the crystal structure with the bound ligand, we find that there is a greater than one value, meaning the volume near the correct oxygen is greater than the volume near the other incorrect oxygen. So the binding preference for all the terpene synthesis we looked at had a larger volume near the correct oxygen. In some cases, there was a huge difference. In some cases, there was a minor difference. Okay, so we see that the enzyme architecture is bound to select how to bind the substrate. So that's the first main finding. Now, that doesn't tell us why. So we look more carefully at why this would be. And we calculated the electrostatic potential in the active sites. Now, these are supposed to stabilize carbocations. So these are highly negative. So this is for the plant one, and this is for the microbial. They look both rather positive, so we couldn't really see any main difference here. But looking simply at the structural motifs, we did find something very interesting. We find that in the plant case, the one oxygen is blocked by a water molecule that is highly conserved and bound by strong hydrogen bonds with nearby residues. So that was clearly that this site is blocked here, and therefore it wants to bind the ligand through this position. In the microbial case, there was a bulky hydrophobic chain in this position which blocks this oxygen and then makes for preferential binding through this oxygen. So we have a good explanation, good understanding why, but we worked on a very small data set. We had a handful of crystal structure where we know the answer. So to increase our data set, we then took about 150 terpene synthase sequences, which are very well characterized, and we know the exact product, and we know where they come from, and they've been well studied. So these come from algae, bacteria, fungi, and so on. And then we calculate the likelihood of having, on the one hand, the plant-like binding motif versus the bacteria-like binding motif. And if we take, for example, the case of bacteria versus plants, so if you see in bacteria, they have a high probability, near 1, to have the bacteria-like binding site, and a low probability of having a plant-like active site. Whereas the plants have the opposite, a low probability of a bacteria-like active site motif and a high probability of having a plant-like. So what we saw in a handful of crystal structure seemed to be consistent with a much larger set of terpene synthase sequences. So that was interesting, and that was good. So when you then go ahead and look at the taxonomic connection and look at the phylogenetic relationship, you see that the plants form a separate branch from all the other non-plant enzymes. So this then explains what we just saw, that plants bind or sense the substrate in a certain way, and non-plants do it in a different way. And the key to our finding here was, of course, initially through our EnzyDock studies. And of course, this is then reflected in the sequence analysis. I'm not going to go into details here, but that's I would say, a key finding, that this is evolutionary rooted, and plants and microbes do things differently. So if you then take and conclude this part of the talk, we then come from the sequence level and this terpene synthesis, which are real magicians in our eye. This is our magician mascot for the terpene synthesis. At the sequence level, we get a divide between non-plant and plant terpene synthesis, which bind the substrate differently and thereby form products in one or another group. Now, what's the meaning of this? How does this insight help us? Well, we don't know quite yet. This is just the beginning, and I think we open up a playfield here. So that's that. I think, in our opinion, that's exciting. But what do we have for now? Well, we have a rule for modeling all terpene synthesis, and I remind you, it's a huge family. So we now know how to dock the non-plant and plant TPSs. We know how they should bind. This can then help us design novel new enzymes using enzyme design methods. And then the key question that came up during the study then was, can we use this to predict the stereochemistry of natural products? So this is a huge question in enzyme catalysis. Do we understand why different enzymes form different enantiomers? So let's take it. So this is then the big question that we're puzzling and thinking about these days. So let's just bring you an example. So here we have the case of biosynthesis of a product called 1,8-cineol. That's this product here, by the enzyme cineol synthase. So if you look at this molecule, see there's a symmetric plane here. And this symmetry makes sure that this molecule is not enantiomeric. It's achiral. However, clever experimentalists have done isotope labeling and shown that the intermediates in the two reactions are of opposite chirality. This is R and this is S. So this one is from plant, this one is from microbes. So when we do the docking of this ligand here and here into the relevant crystal structures, we see a clear preference. The plant binds via the one oxygen, microbes bind via the other oxygen. So our bind or terpene sensing rule is kept up here. So that's good for us. And then if you look at the actual active site, you see that asparagine or glutamine asparagine residue is positioned in flanking positions. In plant, it's here, and in microbes, it's here. And this one has been suggested in the literature to take part in the deprotonation of the final carbocation. So this could then show you how different chirality is induced by the architecture of plant versus microbial enzymes. So this is something that we are actively thinking about. It's not just in this enzyme. It's a frequent observation in nature that chiral products or intermediates of plant and bacterial enzymes are often opposite enantiomers. So this is a general finding. We think we might have here a partial explanation to this phenomenon. So that's something that still needs a lot more work, but we are looking at that actively these days. So okay, so there's a question about whether we considering taking EnzyDock towards more high throughput. So let's move then to the next project, and that is benchmarking docking methods for SARS-CoV-2 Mpro. We did somewhat more high throughput, although it's not EnzyDock is not a high throughput tool. Just by virtue of today, we're using QMMM, which is kind of a slow method. So that's not the main, but we can use it in for hundreds of enzymes in a workflow, which is what I'm going to show here. So when it comes to the Mpro enzyme in SARS-CoV-2 virus, we're speaking about proteases, and we're speaking about protease inhibitors. So in the enzyme, you have Mpro, whose job is to break polypeptides into functional proteins. So without this breaking apart of these polypeptides, the virus can't grow. Now, if you then develop an inhibitor to this protease, of course, then you will keep these polypeptides and they will not break into functional proteins, and thereby you have inhibited the virus replication. So that essentially is the idea being this protease inhibitors. And for this study, we needed crystal structures. And luckily, because of the magnitude of the SARS-CoV-2 pandemic, there were lots of researchers working on this. And already in January 2020, the first crystal structure was published for Mpro. Here we see Mpro, the dimer. And Pfizer developed Paxlovid as an antiviral drug that went from clinical test to in 12 months, which is really, really fast. So here you see the Paxlovid molecule, which is a strong inhibitor for SARS-CoV-2 Mpro. So we wanted to do a benchmark study because we saw that people were doing a lot of screening studies against the Mpro enzyme, and we felt that it's dangerous to do fast screening and suggest people now go ahead and synthesize or test a range of molecules just based on in silico screening, which by itself might not be enough, or should maybe not be enough to convince experimentalists to immediately test them. So that the docking tools might not be accurate enough. So these are the people that took a break from their regular research and delved into this project, which, as with any project, becomes bigger than you expect with time. And for this, we had to develop workflows that will allow us to do this fast and for many different softwares using the same workflow to save time. So the workflow essentially was download all structures from the PDB, prepare them all for docking using the same script, then do, well, I don't know if it's called large scale or somewhat large scale docking, and then data analysis. So we download the structures from the PDB. From all protomers, we removed crystal waters, ions, and co-solvents, split the protein and the ligand, completed missing residues if needed, and added hydrogens to ligands and proteins. And again, we did this not within any of the separate softwares because it would be too software specific. We wanted something that was general for all the software methods. And then if you only had the one monomer in the PDB, we then generated the second monomer to create the correct dimer because the binding site has some interactions from both monomers. So we then set up docking protocols in different docking programs. And these are the docking programs that we tested. We tested Glide, Dock, AutoDock, AutoDock Vina, EnzyDock, and Fred, as these are readily available and commonly used. And we then looked at non-covalent docking, which is available in all of these programs. And covalent docking was not available in all of them. It's available in Glide, in AutoDock, and EnzyDock. And the ligands were fully flexible, and we did this in fixed proteins because this was the redocking project where we tried to dock the ligands back into their original crystal structure to see how well they could reproduce the poses of the bound ligand. Because again, the question is, how well do these programs redock into the original crystal structure? All the data analysis were then done with a range of Python tools, which are commonly used for this purpose, including RDKit. And again, the same protocol was used for all the docking softwares. Then how well do these programs do in terms of docking pose prediction? So we identified correct poses as those having an RMSD of less than 2 angstrom. And we then also tried to see whether if they found the correct pose, which is less than 2 angstrom, is that also the lowest energy pose? So the top part of the columns here for Glide, AutoDock, and EnzyDock is the ability to identify a correct pose. And then the lower bar is whether this was then also scored as the lowest energy pose. So both Glide and EnzyDock finds the majority of the poses, but do not score them as the lowest energy pose. Then if you look only at the covalent bound ligands, not all of them, so some of these are covalently bound, then you see that the success in the docking is higher for all 3 docking programs. And that, of course, is because docking a covalent ligand, because it has an anchor point in the active site, it's thereby easier to get the correct pose. But still, they're not, they don't find all of them, and they're not all scored correctly. Okay, so I might just give a spoiler why this is. The active site in MPro is rather open, and there's a huge desolvation factor here that the docking programs have a hard time dealing with, and there are also specific water molecules that we, of course, stripped from the docking, so they all had to try to do the docking without water molecules in the active site, and that, of course, makes it more difficult. So where do the ligands bind? Well, here you see the active site, which has 4 pockets. It has a pocket called S1, S1 tag, S2, and S4. So these are 3 separate pockets that the ligands may fill all of them or only some of them. And some of the ligands bind in the main pocket, but some also bind at the surface or at the interface between the dimers. So those are, of course, more difficult to predict. as these are not tightly bound ligands. So if you then go ahead and look at the covalent docking and how well Glide, AutoDock, and EnzyDock do when you dock ligands that bind in different sub-pockets. So here you see ligands that bind in 2 pockets, 3 pockets, and 4 pockets. So these ligands are then much bigger than these ones because it has 4 arms spreading into the side pockets. So for all the docking programs, but specifically for Glide and for EnzyDock, they do better for the bigger ligands that fill all 3 pockets, just because the interactions are stronger, and that relates to the idea of the desolvation, which is difficult to deal with. So the better the ligand binds, the stronger the interactions with the enzyme, the less important relatively the desolvation is going to be. Then if you look at all the programs for the non-covalently bound ligands, where we can also include the ones that don't do covalent docking. So none of the codes were able to correctly identify and score ligand poses in more than 26%. So Glide does the best here, followed by AutoDock and EnzyDock. But if we remove the surface-bound ligands, which are difficult to predict, then they all do better. So in this case, Glide is still the best, followed by AutoDock and EnzyDock. So, so predicting surface-bound ligands for MPRO is difficult, or very difficult. But even in the binding pocket, which is what we see, the ones that bind only in the binding pocket, that's also still very or quite difficult to do. So I would say the main conclusion from this benchmarking work is, and that was our message, I think, to the scientific community, is that to propose in silico inhibitors based on docking alone for experimental groups to go try, that might be it might be difficult to predict reliably good inhibitors using docking only. You might need to go further, and by going further, I mean one might need to do free energy perturbation simulations to get free energy of binding. One might need to use additional tools beyond docking. So if I'm going to conclude the talk, so I try to describe EnzyDock, which is, in our opinion, a unique docking program that allows treating a wide range of complex problems in enzymology. And it can dock with quite accurate potentials, including QM/MM potentials. I didn't speak so much about it today, but we have included more informatics and machine learning in EnzyDock in the last couple of years. And it allows us to guide enzyme design projects. Again, I didn't have time to speak about this today, but we have some enzyme design projects where we managed to come up with new enzymes based on our docking program together with enzyme design tools. And it allows easy definition of automated workflows by virtue of being a charm-based docking program. So let me thank my group members, which you see here last year on a trip in the Jerusalem mountains. Let me thank financial support from Ministry of Science, Israel Science Foundation, German Israel Foundation, and the NIH, and collaborators from Germany, Bruck and Loll, and from the Weizmann Institute, Fleischmann. And let me thank you for your kind attention, and I'd be happy to answer any questions.
Yeah, okay. Wonderful talk, Dan. Yeah, we're really fascinated to see how EnzyDock performs. Yeah, so I'll take what's that we are getting in the chat box. So yeah, the first question from Sudeep that you have already answered. His second question, he says benchmarking, especially the docking efficiency, is it dependent on the proteins? Your benchmarking.
Yeah, so
benchmarking.
Yeah, the benchmarking is for sure going to be dependent on the protein. MPRO is a difficult case where, as we saw, all the docking programs struggle. There are other proteins or enzymes where docking will work better. It could probably be ones where it works worse. And I think the bottom line is that one needs to be careful. And probably want to use a few different softwares to compare and do some consensus scoring. Yeah.
There is one question from Seema. For the terpene studies, the plant versus microbe distinction, is it limited to the active site or any long range interactions? Let's say hydrophobic core, flexibility, etc. That differs as well. She's curious about how a chimeric protein of two species would recognize the ligand.
Right. Well, that's an excellent question. So if I understand correctly, the question is whether the binding motifs that I showed in the active site, if there are similar differences further away from the active site. If that's the question, then the answer is we don't know. We haven't looked for those patterns yet. It's more complicated. There probably are. Are those important through long-range interactions? We don't know. I agree it would be really exciting to try create chimeric proteins of two species, and we hope people will try to do this as a follow-up to our study.
Yeah, so I had, okay, so there's one more question from Chandan. For covalent docking, do we need to define reactive groups or it can detect automatically?
Yeah. So it's a good question. Today we need to define it specifically. It's not that it's difficult to suggest reactive groups, but right now you have to define it separately. Yes.
No questions, then I'll take the opportunity to ask questions. Yeah, so I had one question in EnzyDock. So in the recent version, do we take into account for, let's say, dynamics, enzyme dynamics?
Right. So there is some enzyme flexibility. So you can define flexible residues, flexible backbone, but it's not meant to do large scale flexibility, like loop motions is not well described. You'd rather do separate docking for different conformations, or then just go straight to free energy perturbation simulations.
Okay. And yeah, I have another question. That is, can we extend the use of EnzyDock to other class of proteins?
Yeah, sure. You can use it for any class of enzyme or protein. Yeah, it's not limited to. We used it most for terpenes because that's what we have been mostly interested in, but you can use it for any.
Okay. Yeah, there is one more question from A. Satya Sai Kiran. Yeah, the question is, if we have an enzyme which is essentially a metalloprotein, how do we prepare the protein active site for EnzyDock with specific substrates?
Yeah, so then that would be the same as any other setup. You would, I mean, if I was going to do that, I would then probably do the docking with explicit residues that coordinate with the metal, and then include some restraints where you do the classical docking and then refine it using QMMM docking, where you can then remove the restraints because it's treated by QM, and then hopefully the metalloprotein or the metal protein interactions will be well treated. And that's one of the advantages of EnzyDock, is that you can do the QMMM as part of the docking.
Okay. Yeah, so there are no more questions. I would ask our CEO, Dr. Sudeep Roy, to say a few words, followed by Seema to say a word of thanks. Over to you, Sudeep.
Yeah. Hi, hello, Professor Major. So very nice meeting you. Actually, I asked Anil that can I come in as a and meet you, on camera, so that we know each other, get to know each other. Thank you very much for your fantastic talk. Learned a lot, and we work in the similar area, not exactly on enzymes per se, but a lot of drug protein docking, and then followed by molecular dynamics and free energy calculations and things like that. So it's in our bread and butter for the company as well. So and I really learned a lot, and I see that this whole enzyme currently, how the world is going towards more, bringing new enzymes for new molecules and all. This is going to be the future of in silico chemistry and biology, right? So it's great meeting you. Thanks, Anil, for setting this
Yes.
up. Yeah.
Thank you very much. Yeah.
Thank you.
Yeah, now I would like to ask Seema to say more thanks. Dr. Seema?
Yes, I'm here. Can
We can hear you.
okay. I would like to extend a very heartfelt thank you to Dr. Dan Major for taking out time from his, busy schedule to deliver a talk with us at this Fusion talk series. The talk was really enlightening. I personally enjoyed it a lot. And on a personal front, looking at your lab group photo, girl power, I really appreciated the sentiments there as well. But yeah, on a more serious note, thank you so much. Thank you for accepting our invite, joining us, and delivering such an impactful talk on exactly as Shadip described, what is the future of in silico in enzyme design chemistry. With that, I would also like to thank everybody who joined us for this talk. And I hope everybody is leaving this talk more enriched with a lot of information regarding, docking principles, methodologies, and what else to look out for in future. So thank you. Thank you, everybody. Thank you, Dr. Dan Major.
Thank you very much.
Yeah. Thank you, Dan.
Thank you.
Thank you. Thank you.
Yeah.
We'll look forward to see you again, Dan. Yeah. Thank you. Yeah.
Likewise.
Yeah.
Thank you very much.
Yeah.
Bye.
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 Bye-bye.